So welcome back guys. Today we will discuss about gene duplication. So what is gene duplication? So existing gene can be duplicated to create a closely related gene within a single cell. It replicates to form a closely related gene within a single cell, and there are several processes which can lead into duplication. So this can be unequal crossing over and replication slippage. Okay, and duplicated genes may have two fates. So what are the two fates? In most of the cases, duplicated events, they are followed by loss of function mutation, which functionally restore the one gene state that preceded the duplication. So duplication will eliminate the loss of function, and it will help in gaining that function which it was already there. In the alternative fate, duplicated gene remains functional and diverges their sequence and pattern of expression and the taking on different roles. So in the second case, roles are changed. So repeated round of this duplication and divergence over many years, it enables one gene to give rise to a whole new family with related function within a single genome. Example, the globin gene family, it provides a very good example of how DNA duplication generates new proteins. And globin genes are kind of myoglobin, hemoglobin, alpha beta hemoglobin. They are all descended by duplication and divergence from the ancestral gene. So gene A, when it shows gene duplication, it have two fates, one with related function and another with no function. What are gene families? So most of the protein coding genes, they are represented only once in the haploid genome and are thus termed solitary genes. Only once, then solitary genes. But duplicated genes constitute the gene family, which is defined as a group of genes of similar or identical sequence that code for similar or identical proteins. Okay, these are clustered or dispersed around the genome. So it can be simple multigene family or complex multigene family. So what is simple multigene family? Simple multigene family is where all the members have identical or nearly identical sequences which are simple. Okay, and the rRNA genes are examples of simple multifamily genes. So within a tandem array of rRNA genes, each copy is exactly or almost exactly like all the others. rRNA genes are example of simple multigene family. And each cell contains a multiple copies of the rRNA genes that code for ribosomal RNA. So rRNA genes, they are in multiple copies in each cell. So like example, E. coli also contain multiple copies of rRNA genes. It contains 7 copies of its rRNA genes, and human cells contain about 200 rRNA gene copies per haploid genome. Okay, and the multiple copies of conserved rRNA genes on a given chromosome, they are located in tandemly repeated manner. Okay, many rRNA copies repeated in a tandem manner, and they are needed to meet the cellular requirement for their transcript. But what about complex multigene family? Here they are although similar in sequence but are sufficiently different for the gene products. Okay, the products are different. So one of the example of this family is mammalian globin gene. The globins are the blood proteins which combines to form hemoglobin, and each hemoglobin is made up of alpha and beta globin. So alpha globin are coded by multigene family on chromosome 16 and beta on chromosome 11. And alpha subfamily contain 3 genes, zeta and 2 copies of alpha. Okay, alpha subfamily contains zeta gene which is expressed only in embryonic stage, and 2 alpha genes which is expressed in fetal and adult stages. But beta globin gene, it is longer than the alpha gene cluster and contains 5 genes, alpha 3, beta 5. So of the 5 genes, 3 are expressed prior to birth, and the epsilon gene is expressed during embryogenesis, while the other 2, which are nearly identical, known as gamma genes, they are expressed only during fetal development. Okay, so before birth, alpha is not expressed, as you can see. It is in the embryonic stage, fetal and adult stage, but in case of beta, it is 2 which are expressed before birth, epsilon during embryogenesis, and gamma which are expressed during fetal development. And the remaining genes, delta and beta, are expressed following birth. Okay, so epsilon and gamma, they are expressed during embryogenesis and fetal development. Embryogenesis, so before birth is fetal development and embryogenesis. Okay, so in fetal and embryogenesis, zeta and 1 alpha gene is there, but in case of beta globin, there are epsilon and gamma genes which are expressed before birth, and delta and beta, they are expressed following birth. Okay, so 2 gamma and 1 epsilon before birth, and 1 delta and beta after birth. So alpha globin gene family on chromosome 16, beta on chromosome 11. Alpha gene family contains 3 genes, alpha and 2 alpha and 1 zeta. Okay, and here it contains 5 genes in beta, which is epsilon and 2 gamma before birth, and beta and delta after birth. So there are several duplicated globin sequences that are not functional genes. Okay, they are example of pseudogenes, and they have close homology to the functional genes but have been disabled by mutation so that they are not expressed. 2 functional pseudogenes present in the alpha cluster and 1 functional pseudogene is present in the beta cluster. You can see the psi which is there, 2 pseudo is in alpha and 1 pseudo is in beta. So these are pseudogenes which does not express but are present and are similar to the functional genes. Next is homologous genes. So what are homologous genes, what are orthologs, what are paralogs, that we need to understand. So homologous genes are the ones that share a common evolutionary ancestor. Ancestor is common, and a pair of homologous genes have sequence similarity, but they do not have identical nucleotide sequence. Similarity is there but not exactly identical. Okay, it occurs between the two genes, undergo random changes by mutation, and homologous genes fall into two categories, paralogous and orthologous. And homologous genes present in the same organism are said to be as paralogous, which is a result of duplication. And when homologous genes are in different organism, it is said to be as orthologous. So example, myoglobin and beta globin genes of the humans in the same organisms, they are paralogs, and they are originated by a duplication of an ancestral gene. Whereas myoglobin genes of human and chimpanzee species changed, they are orthologs. So gene A and gene A, here you can see when speciation event is there, it is ortholog, and when duplication is there, it is paralog. So if we understand more about pseudogenes, so they are functionless gene variants that are present as a result of some incident, some accident. Okay, and they tell us about the changing nature of the genome. There are two types of pseudogenes, conventional and processed. Conventional pseudogene, a gene that has become inactive, non-functional because of accumulation of mutation. The globin pseudogene are the most common example of conventional pseudogene, and the human globin gene clusters contain 5 pseudogenes, which is okay, fine. And processed pseudogene, that results from integration into the genome of a reverse transcribed copy of an mRNA. What does that mean? That some integration is there into the genome of a reverse transcribed copy. mRNA is reverse transcribed and integrated. So processed pseudogene, it may integrate into the same chromosome as its functional parent or into a different chromosome. So reverse transcript is added. Okay, and this may be in the same chromosome from where it originated or into the different chromosome. So because a processed pseudogene is a copy of an mRNA molecule, it does not contain any introns because it is reverse transcribed, and it also lacks promoter sequences present in the upstream of the parent gene, and the absence of these sequences, it inhibits the transcription initiation and makes pseudogenes inactive. So promoter is not there, introns are also not there, so that they cannot initiate the transcription of its own, and they are inactive. So one is conventional, one is pseudo. Next is organelle genome. So we talked about organelle genome earlier also. So like in mitochondria and chloroplast, they contain independent DNA genome. Okay, genes which are not residing in the nucleus, they are said to be as extranuclear genes. And organelle genome, they are circular molecules, which is not always compulsorily. They are double-stranded DNA which code for some but not all of the proteins found in the organelles. They code only for some proteins, and most of them, their demand is met up by nuclear proteins. And so these mitochondrial DNA and chloroplast DNA, they what they do, they import some of them from the nucleus. Okay, so most of the proteins, they are encoded by nuclear DNA and imported into the organelle from the cytosol after they are synthesized on cytosolic ribosome. And mitochondria and chloroplast contain multiple copies of the organelle DNA genome, and for each human, mitochondrion contains about 10 identical molecules. The prokaryotic character of the organelle genome suggests that mitochondria and chloroplast, they are the relics of free living bacteria that formed a symbiotic association with the eukaryotic cell. So it says that mitochondria and chloroplast, since they are independent replicating molecules, so they might have come from prokaryotic organisms, and this is like the mentioned in endosymbiotic theory. Next is mitochondrial genome and chloroplast genome. So mitochondrial contains multiple mitochondrial molecules, and total amount of mitochondrial DNA depends on the number of mitochondria, size of the mitochondria, number of mitochondrial molecules per mitochondrion. Okay, and this mitochondrial DNA, this is encoded by rRNA, tRNA and some mitochondrial proteins, and they are synthesized on mitochondrial ribosomes, not cytosolic ribosomes. Okay, and human mitochondria contains around 16,000 base pairs which lack introns and contains long non-coding sequences, LNCs. So unlike human mitochondrial genes, some plant and fungal mitochondrial genes contain introns which must be removed by RNA splicing. Human mitochondrial encodes the two RNAs found in mitochondrial ribosomes, the 22 tRNA used to translate mitochondrial mRNA, and 13 different polypeptide chains. So mammalian mitochondria, which does not contain intron, they are transcribed into single transcript from a major coding strand, and individual products are generated by RNA processing. Okay, not all mammals but humans does not contain introns. So the mitochondrial genome is much larger in yeast, and the genes are more widely spaced than in human mitochondrial genome, and some of the genes have intron in case of yeast. And what about chloroplast genome, which are circular molecules of double-stranded DNA, and ctDNA, which is chloroplast DNA, its large length is around 120 to 190 kb, and they contain multiple copies of organelle DNA, typically 20 to 40 in higher plant. And chloroplast genome codes for all rRNA and tRNA as well as some proteins. So that was organelle genome. Next is human genome, which consists of the haploid genome contained is 3.2 to the power 10 to the power 9, and majority is euchromatin. Human genome is around 30,000 genes, and the total human gene number is much less than we had expected, and previously it was expected that it might contain 1 lakh genes, and the average human gene is 27 kb long, and gene occupies 25% of the human genome, and protein coding sequence are a small part of this fraction. So size of human genome is 3.2 into 10 to the power 9, number of genes is 30,000, largest gene is around 2.4 into 10 to the power 6, mean gene size is 27,000, smallest number of exons per gene is 1, largest can be 178, largest exon size can be 106, mean exon size is 145, percentage of DNA sequence in exons 1.5%, percent of DNA in high copy repetitive elements is approximately 50%. Highly repetitive genome is there in humans. Next we come to Human Genome Project. So it was an international government and private sponsored project which was used to map and sequence the entire human genome, which was first formulated by US Department of Energy and NIH, and their goal was to discover and map the 35,000 human genes and make it accessible for further biological study. And it is also conducted parallel studies or genetic map. Also, study was done on yeast, fruit fly, which is Drosophila, mice, Arabidopsis, and after years of multi-billion-dollar research, this was successful and jointly announced draft of the human genome sequence in 2000. And by mid 2001, scientists associated with these ventures had presented the true nature and complexity of the digital code inherent in DNA. So we now understand that there are 35,000 genes in human nuclear DNA, which is around 3 billion chemical bases arranged in precise sequence. So what are the salient features of human genome? It contains around 3,164 million nucleotide bases. The average gene consists of 3,000 base pairs. Gene code for protein dystrophin consists of 2.4 million base pair, longest one. The total number of genes estimated is about 30,000 to 35,000. Size of the chromosome is 55 MB to 250 MB. Chromosome 1 has maximum number of genes 2968 and Y has the fewest 231. 98.5% genome is non-coding. Only 1.5% of the protein is coding sequence. Repetitive sequence make up a very large portion of human genome, comprising less than 10%, and transposable elements about 50%. Interesting, right? So next is E. coli genome. E. coli genome comprises single main chromosome and plasmids made up of circular double-stranded DNA with a homogeneous distribution of genes, and they contain around 4 to 8 actual and proposed gene coding sequence. 88% of the genome encodes protein. Okay, 11% are for regulatory function, less than 1% are for repetitive DNA sequences, and the average distance between gene is 120 base pair. Yeast is having 16 linear chromosomes, each containing a centromeric region required for chromosome segregation. The nucleotide sequence is around 12,000 kb of DNA, and 585 potential protein coding genes and 455 RNA coding genes are there. 70% is protein coding. So unlike most other eukaryotic genes, only about 4% of 6,000 yeast genes have introns, and even then most only contain a single intron. So humans have maximum non-coding region, coding region is very less. Next we come to eukaryotic chromatin and chromosome. So in these topics what we are doing, we are just recalling some concepts, and since this book is very helpful for CSIR net preparations, and so I am reading out from the book, so that even if you revise the book twice or thrice, the chances to clear all the basic concepts, then you can follow some good reference books for particular units, and this will help you to enhance the knowledge, and diagrams in the notes will help you to retain the concept. So this is just the basic we are discussing, which will help you, give you the overview, like how much you have to study, in what way you have to study. Okay, from basic to advanced, everything is given in this book. So next is eukaryotic chromatin and chromosome. So first of all, what is chromatin? Chromatin describes the state of nuclear DNA and its protein. Okay, DNA plus protein during interphase between mitosis of the eukaryotic cell cycle. So in interphase nuclei, chromatin appears to be attached to the nuclear matrix, which is a proteinaceous structure. So DNA sequence attached to the nuclear matrix are called matrix attachment regions, and they are usually AT rich but lacks any consensus sequence. Okay, DNA is attached to the nuclear matrix are called matrix attachment region, and they are AT rich but lack any consensus sequence. And a chromatin DNA molecule contain 3 specific nucleotide sequence: centromere, telomere, and origin of replication. Chromatin consists of 3 regions. Chromatin are attached to the matrix by matrix associated regions, which are AT rich regions. Centromere is the primary constriction site, which is a region of eukaryotic chromosome, whereas a kinetochore is a region where sister chromatids are held together. Okay, and this kinetochore are present in the eukaryotic chromosomal region, centromeric region, and the 2 copies are held together at the centromere. Okay, and the site where mitotic spindle attached to the chromosome during mitosis. Okay, this is the region, kinetochore is the region where the mitotic spindle fibers attach to the chromosome during mitosis and helps in proper segregation of the sister chromatids. And the centromere has no defined DNA sequence. It consists of tandemly repeated DNA sequence. They are made up of 171 base pair repeating unit and are known as alphoid DNA. Okay, centromere is made up of alphoid DNA. Sequences are not fixed, and in yeast the centromeric region is around 110 base pair long, consists of 3 type of sequence element: CDE1, CDE2, CDE3. CDE1 is 9 base pair, 2 is greater than 90%, and 3 is 11 base pair. Okay, 1 is 9 base pair, 2 is greater than 90%, 3 is 11 base pair. And centromeric DNA, they exist in heterochromatin state, which is not expressed, and being them in heterochromatic state is very essential for its function. And when the DNA are duplicated in S phase, 2 copies of each replicated DNA remain tightly bound together known as sister chromatids. So try to understand the duplication in the S phase. So 2 copies of each replicated DNA remain tightly bound together as 6 identical sister chromatids. So sister chromatids, so DNA, 2 copies of replicated DNA, they are bound together, okay, at the centromere as as identical sister chromatids. So sister chromatids are glued together by a protein named as cohesin, and this cohesin, which are present along the entire length of the sister chromatid, okay, when the DNA is replicating. And this cohesin is very important, okay, and this when there is segregation, this cohesin has to be broken, and when there is segregation, that is during mitosis, okay, during late mitosis and at the start of the anaphase, this cohesin has to be broken down to allow the sister chromatids to be pulled apart. Okay, so sister chromatids are where the DNA is in duplicated stage in the S phase, bound together at the centromere, and the sister chromatids are glued together by multi-subunit protein called cohesin. And cohesin are broken down in early anaphase and late mitosis. Okay, so where the centromere is present will tell about the different types of the chromosome. So chromosome can be meta, acro, telo, or monocentric. Metacentric means when the centromere is at the middle. Acrocentric, centromere is at the end. Near end is acro. Extreme end is telo, where telomere is at the end, so telocentric chromosome has the centromere at the end. Monocentric means single centromere. Holocentric, multiple centromeric domain. And metacentric means in chromosome that is not metacentric, means one arm is bigger and one arm is smaller. In that case, P represent the short arm and Q represent the long arm of the chromosome. You can look at the picture somewhere, okay, because here it is not given. Coming to telomere, so this telomere, they are specialized structures which cap the end of the eukaryotic chromosome present in eukaryotes, and they maintain the structural integrity of the chromosome. So if telomere is not there, the chromosome will become unstable and ensuring complete replication of the extreme end of the chromosome. Telomere ensures that the end, up to the end, the replication occurs. And this telomere, they consist of long array short tandemly repeat sequences, which may be 100 to 1000 repeats depending upon the organism. And one unusual property of the telomeric sequence is the presence of GT rich single strand, usually 14 to 16 bases. Okay, GT rich single strand is there, which is 14 to 16 bases, and the GT rich sequence, it is generated because there is a limited degradation of the CA rich strand. Okay, because CA is there, that's why GT rich sequence is also generated. Okay, and the property of the telomere is GT rich. So the sequence of telomere, it is highly conserved in evolution, and there is a considerable similarity in the simple sequence repeat. For example, so GT, you will see it's moreover similar in different species. It is TT, GG, GG, Paramecium, TA, GGG, Trypanosoma, TTA, GGG, Arabidopsis, TTA, GGG, Homo sapiens. Okay, so guanine bases, they have the unusual capacity to associate with one another, and 4 guanines can form a planar G tetraplex. So 4 bases hydrogen bonded to each other. Okay, and this structure describes the unusual property of G rich sequence because G tetraplex will be formed. So how this telomere is responsible for the stability of the chromatin ends? So there is a novel structure called T loop found at the end of the mammalian telomere, which is responsible for the stability of the chromosomal ends, and this loop is formed at the end of the chromosome. When we will study about DNA replication, we will understand about telomeric replication as well. So this T loop will be found at the end of the mammalian telomere and is responsible for the stability of the chromosomal end. And this loop forms at the end of the chromosome under the direction of a protein called TRF2, telomere repeat binding factor, which is present at the end, which forms the loop. And this loop causes the 3' end of the chromosome to loop around and interdigitate into the double helix, forming the loop. Okay, so this TRF, telomere repeat binding factor, it helps this 3' end to interdigitate and loop around into the double helix, forming the loop. And this is how the chromosome is replicated up to the end, and the average length of the loop in animal cells is 5 to 10 kb. Okay, so next, this is how we are proceeding towards the replication. So this is the basic we need to know before we start to understand about the replication, transcription and translation. So from the basics of what is gene, number of genes in E. coli, what are exons, what are introns, what are repetitive sequences, this will build up our base to understand about the fundamentals. So origin of replication is what? Is the region where chromatin contains ORS region, origin of replication region, not ORS, it is ORI, and ARS region is present in yeast, autonomous replicating sequence, which is the origin of yeast. Now here ORI region is there in the chromatin, it's the origin of replication, and individual replicons, they are relatively small, which is 40 kb to 100 kb, and in human beings replication requires about 30,000 origin of replication, with each chromosome containing several hundred ORI. Okay, and each origin of replication represent a replication unit. Okay, each will having several proteins, several factors associated with it, known as one unit. Next is what is hetero and euchromatin. So you can see in this picture the location of hetero and euchromatin is mentioned. Euchromatin is where the genes are activated, genes are loose so that they can be expressed. Heterochromatin is where genes are present in kind of tandem clusters which are not expressed. Okay, so scientist named E. Heitz, in 1928 coined the term U and heterochromatin to describe the part of the chromatin that are less coiled and those that remain highly condensed. Euchromatin are less coiled, hetero are highly condensed. Euchromatin is the light staining, less condensed portion. Less condensed means genes are open, more transcription factors can bind and the genes can be expressed. But heterochromatin is darkly stained because the genes are condensed. Okay, and this condensed state during the interphase, this heterochromatin is believed to be transcriptionally silent because of the condensed state, and different cell types contain different amount of heterochromatin. There are two types of heterochromatin. One is constitutive, another is facultative. Constitutive heterochromatin is where condensation remains throughout the cell cycle and development, which contains highly repetitive sequences and present a role in a chromosomal structure. So it is permanently condensed and contains highly repetitive sequences that are not transcribed. Density is very much reduced that are translocated into or near it and are often inactivated. So this replicates in S phase and has a reduced frequency of genetic recombination. But in case of facultative one, the regions of euchromatin are converted to heterochromatin state. Some euchromatin in a specific condition they are converted to heterochromatic stage, and it has a potential for gene expression at some point in development and can be condensed or decondensed depending upon the cell type. Example is mammalian bar body, which is a very excellent example of facultative heterochromatin. So we will start with bar body in the next video.